Good morning, everyone. My name's Erica Donner, and I'm going to be chairing this session this morning. I'm from Australia, from the Cooperative Research Centre for Solving Antimicrobial Resistance in Agribusiness, Food and Environments, and that's part of our implementation plan for our National Action Plan for AMR. Co-chairing with me this morning is Paul George, and he's right here. He's going to take us through the subsequent presentations, but it's my honour this morning to introduce our first speaker and keynote speaker for the session, Professor David Graham, and he's going to be speaking to us about integrated surveillance along the One Health continuum. So David has done a tremendous amount to get everyone in here, all of our work and around environmental dimensions of antimicrobial resistance into the forms in which decision makers can engage with it, and it starts to make impact through policy. So I'd like to just tell you a bit more about David's background. He has led the AMR and other surveillance projects around the world for 30 years now. He co-led the writing of the very influential 2023 UNEP report, Bracing for Superbugs, Strengthening the Environmental Action in the One Health Response to AMR. And he chairs the integration of the upcoming Quadripartite Guide on One Health Integrated Surveillance of Antimicrobial Use and Resistance. He co-initiated the Pan-American Network for Environmental Epidemiology, and he co-authored the 2021 World Economic Forum report on the economics of waterborne AMR. Domestically, he was a member of the Strategic Advisory Group for Emergencies subgroup on COVID-19 transmission in the environment, and he's been in the Expert Advisory Group for who operationalized the UK wastewater surveillance network during the pandemic. David is a chartered professional civil engineer. He has expertise in microbial genetics and environmental chemistry, and this provides him with a broad background suited to integrated AMR studies. He works from hydrodynamics through to metagenomics, a really broad range of topics and expertise. His current interests span the ecology of AMR gene exchange through to identifying research and communication gaps that hinder One Health solutions. So please join me in welcoming David to the stage.
Thank you, Erica. And I want to thank you for the invitation, and especially Dominic and the team for having me provide this presentation. Of my expertise, what I'm primarily going to be talking today is about communications and how we make environmental research relevant to the other sectors in the One Health spectrum. And so my goal is to give a bit of a sense of what we can be doing that will make our work of greater impact relative to the decision-making process. So I'm first going to fairly briefly talk about bracing for superbugs, the conclusions of what the main conclusions from the report, and then talking about what sectoral risk concerns might be. Then I'm going to talk about some work that we've just published to do with what we've been doing for the last 20 years and how it might fit to what we might be doing in the future. And then I'm going to provide you a few examples related from my own experience of where we've almost got integrated surveillance correct. So at the end, I'll provide you some tips about things that might make the work you do a little bit higher impact. So I'm not going to really talk about the report in great detail, except for the fact that this is the Bracing for Superbugs report, except for the fact that it had quite a lot of impact relative to internal processes at the UN. And I think it's not a gross exaggeration to say that this work that was done by a group of us and many other experts who contributed was the reason why the UN moved from tripartite to quadripartite. It convinced the senior scientists that the environment had to be considered in the health decision-making process. And that then led us into One Health, which is basically, it recognizes the health of humans, domestic animals, wild animals, plants, and the environment are so interlinked that we can't solve one problem related to health, for example, without addressing all the other sectors in the same time. And that's what my talk is about. So I'm going to go back to the very basics. And I usually use this slide when I'm giving it to people who aren't as educated as you guys are. But I want to say, change this slide one bit. And this is basically what causes AMR. What causes AMR basically is we expose organisms to chemicals. And unfortunately, what we tend to do as environmental people, we tend to focus on the consequences of that rather than the action of that. And the action of that is actually the antibiotic use step. And that's something that we tend to miss in the studies that we do. It selects for organisms that are potentially more competitive, ecology, and you end up with a slightly increasing number of resistant strains out there. I might note that this is not a terribly technical talk. It's more philosophical and reflections. Just if you want to have big metagenomes and the things, you might as well go, because I'm not going to talk about that sort of thing today. So the bottom line is that we're having this event of the development and selection processes happening absolutely everywhere all the time. How do we go about measuring that in a way so that we can come up with solutions within each sector? So we've got you've got overlapping pathways with variable exposure effects. I personally believe that the use of antibiotics is the primary problem. Environment is part of the problem, but basically environment is a place where spread predominantly occurs and maybe a tiny bit of transmission. So the use is really its central focus, and actually that was one of the main conclusions of the Bracing for Superbug reports. But it doesn't necessarily mean that immediate use is the only risk. And for you risk assessment people, I apologize, because I'm going to grossly simplify what you do. I highly appreciate all the technical detail, although I don't understand most of it. But there's two risks that we're trying to prevent. This is through our integrated surveillance. The first risk is we want to reduce the possibility of a human, animal, or a plant being exposed and affected by a drug-resistant pathogen. So it's the immediate effect. This is at the bedside. This is the bedside consequence. And the bedside consequence is the thing that the other sectors care about. So we got to bear that in mind when we're doing an environmental study. That's really critical. The second side is what we're really good at, and that is the evolutionary development of resistance and its movement around the environment. This, we know, is increasing the prospective gene pool of potential resistance, but it's not as immediately important to the people who are our primary clients, which are the people who are working at the bedside or with the chicken in the farm or the person who's dealing with the plant in the agricultural system. So when we think about what we should be using in integrated surveillance, we've got to really think about what we should be measuring. What is the information? The measuring is our way of communicating what we learn to other people. So the first and most primary thing is living AMR pathogens. We need to know AMR antibiotic use, antimicrobial use, and we also have to collect metadata, and I'm going to talk about this. And these are my top priorities. I'll explain why the metadata in a little while. The next thing in my list is non-pathogenic organisms that could potentially be the donors of potential resistance to the pathogens. That's my second priority. And these are all living organisms, because those are the things that people in the medical, veterinary, and crop communities care about. But we know that the genes, mobile elements, are also important. I put them third. But it doesn't necessarily mean they're not important, but we have to understand that we've got to do that work within the context of the first two priorities. And I'll get into that in a little while. And the final thing is collecting the other types of things that might influence selection, which is the presence of in situ microbials, the heavy metals, and other chemicals in the environment that might nudge selection or development a particular direction. So, getting back to the UNEP report, based on these general recognitions that I've just said, I want to the UNEP report, and bear in mind that I'm not talking on behalf of UNEP. These are all my own opinions. That the first and most dominant thing that's important is the issue of dealing with untreated human waste and promoting WASH guidance. I say untreated specifically because I'm actually quite a big fan of wastewater treatment plants. I don't think people realize how important they are relative to the health of our environment. I mean, when Jerry Wright's first talk, he showed this incredible reduction of infectious disease. And it's because of the fact that we got antibiotics. However, it's also because most of the world has got wastewater treatment plants that doesn't have high levels of infectious disease. So it's not just the antibiotics, it's the fact that we've got those treatment plants. They may not be perfect, but they're certainly a whole lot better than nothing. And that's part of the reason why I work mostly in emerging and developing countries, because they're the places that have got the real problem, because they don't have wastewater treatment plants. So I'll get off my bandwagon, and I'll go back to the. So the next part, and this is something that Anthony So's I'm sure going to talk a lot about on the talk on Friday, is that we're concerned about the other things that we do that could promote the potential spread of resistance, and rather than particular things, we're actually concerned more with the value chains, the value chain to do with healthcare, the value chain to do with agriculture, the value chain to do with pharmaceuticals. And that means that we should be concerned about the whole pathway by which an original active agent eventually ends up in a person. So that whole thing, so you can't arbitrarily poach steps of the value chain and not have it affect other parts of the value chain. And this is something that I don't know whether Anthony's here, but it's something Anthony taught me, and I've now changed a lot of the way I think because of Anthony's guidance and knowledge. So when it came to the bracing for superbugs, our main conclusion is managing AMU is key to preventing AMR. And integrated surveillance is key to prioritizing how we come up with solutions. But that varies across sectors, and we've got to make sure that we communicate across sectors when we're going about trying to address that. Now, the report was, as I said, I think it wasn't perfect, but it was really successful because it turned a lot of heads and convinced a lot of different types of thinkings at levels that made a difference relative to what we do. One of the consequences is the UN put together the Quadripartite Technical Group on Integrated Surveillance on AMU and AMR resistance, and the mandate for that is to answer the so what question. Okay, we can measure all these things, but so what? Environment is now part of this. We're quadripartite, we're not tripartite anymore, but does it matter? So we've got to start thinking a little bit different because we've got to fill that so what question so that the people, say, in the medical community or in the veterinary community can use the information that we are actually generating. Without going into anything, the guide is currently embargoed, and we're going through the final revisions. There's 4 things that are in the guide. First is a review of current approaches. What have we done so far? Second is, how do we harmonize what we call things and how we do things? As I'll show you in a moment, different sectors do things really differently. And if you generate what you think is glorious data, it might be useless to somebody else in another sector. Third thing is you've got to clarify context. And this is functionally, I've got a long list of things there, but context, I mean, is generating the data that allows you to compare things from sectors. So one might be a stool sample, one might be a soil sample, one might be a wastewater sample. Somehow, you've got to have those different pieces of information harmonized in a way that you can make decisions. And finally, we have to come up with the suitable indicators. And to be honest, the first 3 are fairly easy. The last one, I think, is the hardest. And it's the one I've been least involved in. And I've been trying to avoid it because it's so difficult a decision. There just isn't any black and white answer. I'll talk to other people who know more about the details and ask them. So this is the important thing. Now, the part of this process of generating the guide, myself and Yang Guangzhou were responsible for looking at the past. We're old. We looked at the past pretty good. We were in the past. And so, but we had some brilliant, absolutely brilliant PhD students. I was working on it in Newcastle. Yang Guan was working it at the Chinese Academy. And he had 2 outstanding students, Kai Chen and Shilo Li, who were working on this from an artificial intelligence perspective. And what they were doing was something like I couldn't even imagine. And we published this paper. It came out about a week and a half ago. And although I was on it, I do believe it should be required reading for creating impact because it tells you what we've done. And I'm going to highlight a few things. Kai is going to be talking 15 minutes after I talk, unless I talk way too long. But I'm really hopeful that Kai will be talking, and she'll tell you about the details of what came in this work. I'm just going to highlight a few things for the purpose of my grander thought. First is, what Kai and Shula did was they used artificial intelligence and natural language processing to examine a mere 254,000 publications from 2003 to 2023. It's not 100% up to date, but the bottom line is that we're creating a living document that's going to be publicly accessible. We mapped research integration, methods, what's happening in sectors. We mapped everything based on what we did in the past. And what we see is some promising things. We see on the right, if I don't screw this up, we see that there's exponentially increasing research levels. Doesn't necessarily mean it's integrated, it just means it's research. Obviously, people are doing more and more AMR research, but what's really important is it's not being done in an equitable and equal way around the world. There's almost nothing that's being done in the lowest income countries. Lots of things in low to middle income countries, but not the lowest income countries. And that's where I think a greatest amount of work will be needed, and to be honest, that's where I'm going to be focusing a lot of my efforts in the future. But we're getting better. So if you look at the bottom graphs, this is before 2011, 2018, 2023, and whether it's a red box or not tells you whether there is it gives you a measure of the level of research in that sector, and then it tells you interlocking research between sectors. So what we're seeing as time is going on, and this is based on what we're doing, that there was almost no integration of work. There is now a little bit, but in the last 3 or 4 years, it's got quite substantial. And there's two possible reasons. One is that's where the money is, and we're writing grants that are a little bit more comprehensive, but I think it's actually partly due to the pandemic. Because the pandemic has taught the people who will give us money that the environment does matter. And in fact, we're probably getting, it's sort of a reciprocal thing, is the pandemic has changed a lot of the mentality amongst the people who are making decisions. And they're more willing to listen to the fact that we are doing things. Now, as you can see, not all the boxes are bright red. That means that there's not as much integration, real integration, as meets the eye. But at least it's getting better. And I want to highlight two things that are really important. And the first thing, and this is possibly what I personally think is the most important thing for this audience. This is a confusing matrix that shows you different methods used in AMR research and the relative levels of that research over the last 20 years. So there's some things that people have been doing for the whole 20 years. If it's red, it means there's a lot. If it's blue, it means there's little. But there's some things that we're still not doing. And that's the most important one, to my mind. And you probably can't read it, but basically what it's isolate sequencing that we collect for samples from the environment. The only way we can contextualize what we've measured in the environment is by isolates. Now, we know that's an imperfect thing because microbiology is microbiology, but the bottom line is we just simply have not done it, and it is now one of the things we must do if we want to start trying to make our work of greater value. The other thing that I want to note is there's integration sectors where there's very, very little integration. And those are related to plants and food. Now, why is that relevant? First is, for those of you that, especially on the ag side, there's a lot of opportunity because there's no competition right now in those two sectors. But what's important is the work we've been doing in India and Bangladesh and other countries that have got, particularly in slums, which is where we work, the predominant pathway by which AMR is spread is through food. It's not through water. The people who live in those places know their water's bad, so they don't drink it. They aspire to drink bottled water. But the bottom line is, then they boil it. They do all different things to protect their water, but they don't realize their food is getting contaminated in the wholesale markets upstream of where they're actually getting their food. And the AMR, and we've got pretty unambiguous data that we haven't published yet that shows that this is the case. So you have to wait for about 6 months because I'm a bit backlogged on papers. But that's really important. And there's some good work out there now that shows this. So that's my background. So how am I doing for time? 9 minutes left. Yeah, all right. Well, I'll do my best. I'll talk as quickly as I can. This is my what I've called this efforts at almost integrating AMR surveillance. So it's my own experience of where we. So bear in mind. So I'm sort of a bit, because of the type of. I've got sort of access to talking to people across sectors, and I sort of have a bit of a heads up of what's likely to be important. And so we've tried to become more integrated for quite a few years now. So my first example of my almost successes was the work we did in Malaysia. So the other thing about this is that we work around the world. And one of my examples in Malaysia, one's in the UK, and one's in Bangladesh. So we've got to be thinking holistically to provide the type of information that people that care and make decisions can use, but have it be cross-sectoral. And so this was work we did on the Skudai River. It was primarily done by Emily Ott, who was an exceptional PhD student I had. We published a couple of papers on it. Basically, what we attempted to do in this study was to do an integrated One Health assessment of a river in Malaysia. So we measured water quality, we measured volumetric flow rates, we measured wastewater treatment inputs along this catchment. We were aiming to develop models. We monitored in situ antibiotics. We had an agreement with the local health authorities to provide us AMU data that we could complement with the AMR data we were doing in the environment. And then we also did culturing, genetics, and genomics. So we got incredibly good quality antibiotics data due to Karina Jin and her great team. We had resistomes, microbiomes, isolates. We did all the things that I just said that you're supposed to do. And we found it in a really, really good part of the catchment, which was the upper two sites in the catchment. And what's significant about this area is this is the pristine-ish site, and this is a site that's downstream of a major subdivision of Johor Bahru. And this is the difference. That's what we find in terms of resistance genes here. And this is what we find in terms of resistance genes here. So the idea was that we had this ideal place where we could actually integrate One Health information in a very isolated way where the results were kind of black and white. It was human waste. It was human waste getting piped untreated into the river. So we thought we really had it. But the government, because they saw the data, decided not to give us the health data. The data was so incredibly damning of the quality of water in the river, all the cooperation that we had vanished. So we had a really, really good observational data that didn't really fit the integrated health endpoint requirement. So we were doing other things, and Anthony asked me to include some of our wastewater epi work, those other people here that have done much more than we have. But at the very end of the pandemic, my group in Newcastle was asked to look at AMR using wastewater monitoring. And we did a little pilot study that Kelly Jobling, for all intents and purposes, led. Kelly's here, and there's a poster about this, I think, tomorrow that Kelly's going to chair. So if you want to know a little bit more about this, you can talk to Kelly. And Natalie Sin did a lot of microbiology. So we did this in Newcastle. We had very solid funding. The problem was that we had to do it quickly. Everything you did during the pandemic had to be done yesterday. And so we did everything, but we sort of knew what we were doing. And I show this figure because of all of this information. We had 8 sites in the city. We originally had 17. Yeah, good luck. We originally had 17. We narrowed it down to 8, and for the sake of today, I'm going to talk about 6. But we measured flow rates. We got population equivalents. We're getting community-related information that could be translated into what could be useful for somebody else to use the data for some practical purpose. We got incredibly good antibiotic data, and the hospital waste sewer line looked like a hospital sewer line. It's primarily drugs related to UTIs. We did the same sort of thing to do with sewers for microbiomes and resistomes, and this is all sort of too much. But those are our hospital sites. Those are our hospital those are the taxonomic elements that we were finding associated with hospitals, and the hospitals had predominantly had infectious agents, more so than the other in the community. So we thought this was great. The problem was that we did it in such a rush, we weren't able to collect a lot of the other data. But we thought we really had a wonderful way to go about doing studies like this. So we went to the National Health Service, and they could not believe what we could provide them. They particularly liked the antibiotics data, but they also they don't know almost anything about what's happening within the communities. And so we decided that we would do another study. In another study, we're quantifying AMU at the hospitals. We're looking at all different hospitals within the community. We're looking at much more microbial isolation work, which we had done already, but we're going to do more, focusing on escape pathogens. We're gathering wider community data, which the health people are going to provide us, and then we're going to do genetic source tracking and all different other things. So this is an example. If you've done the right thing and you provide the case, people who are in another sector, in this case the health service, will buy it. And they saw value in what we were doing, therefore they were willing to contribute and then provide us the other information, We've just finished this field work. We haven't done any detailed analysis yet, but we're looking forward to having something comprehensive. I was going to say I had data envy of Amy's talk when she talked about their cooperative work with the hospitals. For those of you that saw Amy's talk, there was very interesting stuff related to the type of thing that we're doing here. But I think what we're doing here is at slightly larger scale. So the final example I'm going to give, so I'm going to try and be done in a not too ridiculously long time, is what I think is really, really useful. And this tells you something at the coalface of the health crisis relative to AMR. And just before the pandemic, we did a study on infant health in Bangladesh, in informal settlements in Bangladesh, basically slums. And we looked at, what we were interested in was what was in the AMR in the infants related to antibiotic use, nutritional status, in other words, what their existing health status was, and then water and wastewater exposures in their environment. And this is what we found. This gives you a sense of the environments that they lived in. So our sewer drains were drains like this along the side of the pathways where the dwellings were. We were collecting water supply samples, which was often a bucket by the road sitting beside the drain. So it was a very unsanitary environment for the most part. But what we did was, and because we didn't have a lot of money, this was a pilot study, but it ended up being an incredibly useful pilot study. We did drinking water, sewer drains. We had 73 volunteer contributors. This is their family, the parents. This was with infants between the age of 6 and 24 months. And then we had over 60 metrics to do with their health, and we also had AMU data. Now, the problem with AMU data, and those of you, it's the hard part. It's actually a bit harder than the do it. The AMR we can measure. AMU becomes more of a problem. But in this particular case, we got a cloudy picture of AMU based on talking with the health workers. And about the most dominant antibiotic use in this particular area was azithromycin. But there was also cephalosporin and some fluoroquinolone use. When it came to our cohort, but we also got an estimate of about 60% of the medication use was actually self-prescribed or not doctor prescribed. But when it came to our infant cohort, there were 6 infants that had been exposed to azithromycin within a week of when we did the sampling, 2 with amoxicillin and 1 with erythromycin. All the rest lived in this relatively unsanitary environment. And so that was the best we could do, but I thought it was pretty good. So we found exactly what Mohammed saw in their work in Bangladesh, looking at poultry, humans, and markets, that the human microbiomes were very different than the environmental microbiomes. And we also saw the diversity of microbiomes, this is microbiome data, was much higher than the diversity of what we found in stool samples, which is exactly what they found in their work on poultry farms. We also found really big difference in terms of the carriage of AMR, and in this case, AMR, antibiotic resistance genes. Now, this is kind of a muddy picture, but the big thing is if you look at the yellow boxes, those are all environment. You look at the blue boxes, those are all stool samples. Environment's higher than stool samples in everything except in macrolides. And in macrolides, stool samples are much higher. We'll get to the reason why this is relevant in a second. So even though the environment had much greater diversity and potential, the actual levels in the stool samples were much lower. I'm close. So this was something that was really important to us. And so we did statistical correlations. We tried to do all that. We couldn't find any correlations when we looked at groups of antibiotics versus groups of antibiotic resistance genes and groups in the environment between the stool and the patient. But what we did was further analysis, and this is what Gilles and Louis did, when just recently, and this is also not published yet, but it's going to be pretty close. What we found was kind of astounding, that if you measured correlations based on the trees with the trees, environmental trees with the trees that were prevalent in the stool samples, you didn't get any correlations. But when you sifted through what I said was wrong. When you looked at the forest and compared with the forest, there wasn't a correlation. But when you looked at the trees within the within the rhizostomes versus the trees of the other rhizostomes between the infants and the environmental samples, we did find some very significant genes. And in fact, ERM-F, one gene, was by itself explained about 90% of the relationship between resistance. Now, ERM-F is selected by azithromycin, which is the antibiotic that these infants are being exposed to most frequently. We can't do a correlation because we don't have enough samples. The other thing that was interesting, we found ERM-F abundance was inversely correlated to nutritional status. So the infants that were undernourished had higher levels of ERM-F than the ones that were not undernourished. We can't say that's because of using antibiotics, because it's a very possibility that the undernourished infants are the ones that are more likely to get ill, and they're the ones that are being given the antibiotics, and we didn't have that information. But you can see we're getting pretty close to the coalface. But the thing that's most important to me is that the environment had all this pool of resistance potential, but it wasn't being manifested in the infants. What was being manifested in the infants was related to the actual drugs they were taking. In this case, it was very fortunate that it was azithromycin. If it happened to be meropenem or another type of drug, we would have seen something completely different. And so the key thing for me from this is me stepping out and putting my policy guidance hat on. You got to reuse incorrect use. You got to reduce use, but you've also got to reduce incorrect use to start reducing the potential for spread through the environment. So, I'm not going to be too long. This is the last one, last slide. Okay, what is needed for integrated AMR surveillance for One Health action? So, the future integrated surveillance should be based on so what thinking. Okay? Think about what's going to be used. How, got to come up with a whole purpose for what you do. And these are my guidance. These are my tips. Include microbial culturing, isolate ID, and sequencing of organisms that complement the samples. Now, I've seen some wonderful stuff at this meeting which says we're going the right direction. I saw 2 or 3, there was a couple of talks particularly that I thought were really good for this. But it's obviously a lot of time-consuming work initially. But we've got to do this work initially to start getting the buy-in of people who are thinking from the WHO, people thinking from the FAO, people who are concerned about animal, plant, and human health. Second thing is this is the wrong one. Oh, wait, never mind. Second point was we want to have a quantifiable endpoint in the study related to health. This is supposed to be and measure AMU, whatever happens to be the AMU exposure. And so you want to have that information because it's information relevant to the other sectors. You have to have as much metadata as possible. How can you compare a stool sample with a wastewater sample with a soil sample? Now, Tong Zhang has done some wonderful stuff in trying to integrate how we measure things, but the bottom line is all that other data was needed to say this stool sample in this cow is explained by this mosaic of rhizostomes and microbiomes in a sewage or drinking or source water sample. So you've got to have the data and you've got to have detail. You can't just focus on your own specialization. You've got to work with other people who do other things to provide those complementary things you need. Finally, we've got to promote greater work in lowest income countries. There's a catch-22 there. We want to do things, and there is work going on that's being funded, but it's often unintentionally academic imperialism in the sense that we're going in, do a study, we come away from the study that we've done in whatever country, we've got a great manuscript that's maybe got into Lancet Planetary Health, and the people that we've worked with in the place we've just worked with can't do it again because so we've got to focus on that. We've got to really build up that capacity. And I mentioned this one because I said the quadripartite QTGAAIS, this is to write a guide. And now we want to take things like the guide and use that guide and get it implemented, particularly in lowest income countries, and give them hope. Finally, we want to perform research that answers so what, and so what our clients care about, the other sectors, or you will be ignored. And to be honest, we have been ignored. for the most part, in the work that we do. And it's because we don't answer, so what? Thank you.
Thank you. So to keep things moving, I'll welcome our next speaker. By the way, my name is Paul George. I'm an assistant professor at Laval University, just up the road in Quebec City, and I'm co-chairing today. And I will be welcoming Andrea Ottesen to be giving our next talk, the One Health, One Data, describing antimicrobial resistance in soil water in the United States.
Thanks a lot. This is my first EDAR. I'm incredibly impressed and honored to be here. So I love the concept, One Health, One Data, but I actually didn't get as far along in our own data set to sort of demonstrate this with data, so I'll explain the concept a little bit more at the end of the talk. But first, I will introduce where I'm from. Oh, disclaimer. So I work at the Center for Veterinary Medicine at the Food and Drug Administration, and this is where NARMS is housed. Many of you are probably familiar with NARMS, National Antimicrobial Resistance Monitoring System. And NARMS has a history since 1996 of integrated surveillance of AMR associated with humans and animals. More recently, VetLearn, which is our Veterinary Laboratory Investigation and Response Network, also coordinated out of the Center for Veterinary Medicine, has been monitoring companion animals, cats and dogs primarily, and AMR associated with pathogens of those. Very recently, in 2021, we've started collaborating with EPA to sort of bring in a One Health strategic plan and focus on surface water. Surface water was selected. I mean, a lot of people here are looking at wastewater, which is obviously the most significant for a lot of epidemiological questions, but as an input to food and agriculture, surface water really kind of has a lot of value for our group. Data is all integrated at NCBI, and this gives us a nice kind of One Health portfolio with this pilot project. And these are the agencies involved here. So I'll just a little shout out to this project. Here are the rock stars from EPA that are associated with this project. And I hope I have the right number for Allison's poster, number 16 on Thursday, if you want to hear more about this initiative. And she also just recently published a paper, which is kind of a white paper on this kind of One Health approach to surface water. At the exact same time, I got funding from our chief scientist at FDA because she's super interested in a high throughput approach to monitoring AMR. We focus on surface water just to start as a pilot, but the idea is that the tools will be used for food. We have a lot of complicated materials that are coming through fecal microbiome transplants. You certainly don't want to have an FMT that has, resistance phenotype that's going to impact you. Probiotics. There's a lot of stuff where this becomes very important, and so we want an approach to be able to describe high throughput AMR beyond just like genomes and their phenotypes. Obviously, we'll focus on 4 significant pathogen types, but we can do more. So, and then with this kind of metagenomic and what I call quasi-metagenomic data, you need some kind of consensus or ensemble pipeline where we can be sure if there was this gene there, we have this confidence. So that's one thing that we're working towards with this project. And then we're going to also provide a baseline understanding of the AMR across the country in surface water, as a byproduct, also in collaboration with the EPA initiative. And one thing we often do is we sort of go up and downstream from a point source, and that's a little difficult for stakeholders and industry to swallow at this point, especially since we don't really know what we're doing with that data yet anyway in a policy realm. We sort of stepped back and designed our site selection to, USGS has classified the entire United States with land use. You can use that data to identify the 95th percentile for urban, agriculture, and natural. So we have those 3 foci as we go across the country sampling water. That's what that looks like for my home state of Maryland. And then we overlay this with public access points like boat launches, and then we can get a nice public access to sample these waters. So we are building up our sites. That's what it looks like in Arkansas, Florida. We're about to do California next week. And so we're slowly building this up so that we can talk about, just trends across different components of the environment: waterfall, frost-free, temperature, east, west. A very exciting component of this is sort of the biogeographic distribution of plasmids that's sort of shaking out of these data. North and south is very important as we think about emerging with global warming. And it's also going to be very interesting when we contrast these data to the EPA focused initiative, because it's a very high resolution water scale evaluation, multiple time points, lots of metadata, but it falls within the 95th percentile, so we will be able to sort of contrast these approaches and see what we learn. So to do this, we also need laboratory methods that are going to work. And this is sort of speaking to what you were talking about in terms of having an isolate. This is a really nice hybrid approach that has been absolutely invaluable to FDA for a lot of different reasons over the past 15 years. So we shotgun sequence the enrichments of all of our pathogens. When you start doing this, you will find so many more AMR stories, from just contrasted to just a genome's phenotype and contrasted to just a metagenome. And so we certainly do the metagenome as well, and we do go all the way through at the end, and we have 400 isolates from every site. At FDA, we just kind of commonly call this quasi-metagenomics because it's an oxymoron to call it metagenomics when it's an enrichment, so we were calling it that. Open to new thoughts on the name. It's a little bit kind of just our wheelhouse. But we have used this to examine the enrichment efficiencies of our pathogen detection approaches. We can find serovar biases because, often the serovar that you end up with is really probably part of an ecology, and you're only calling it one serovar, but really there may have been 3 or 4 in there. Also, we can, like for example, the listeria outbreak in ice cream, in 24 hours we could tell you the serovar. That saves 5 days, so that's illness. Illness is avoided. It's also products that didn't need to be destroyed or held or recalled. So this has an incredible value to AMR research, and we can show you that with a little pilot we've done. So this is what that would just look like. If you're doing a validated salmonella enrichment protocol, you're sequencing this along the entire route. And that's going to tell you really different things about the AMR that's there, and of course it's going to tell you really different things about the taxonomy is there, so that's another way of mapping it all together. If you freeze back your BPW enrichments, your general pre-enrichments, or your selectives, you can go back any time and regrow something that's got a plasmid. Very little, low labor to just freeze those back. So we did do a pilot with 2 sites just to see how well this would work for AMR. And we have one site that was just downstream from a hospital and one site that was in a reservoir for drinking water. And with even 100,000 reads, no, 100 million, sorry, shotgun sequence reads, you're not going to find beta-lactams. You're not going to be able to, maybe you'll find a tiny bit if you have really deep sequencing, but you're not going to have enough that you can really make an informed comment about that environment. But when you do like one of the pre-enrichments, you can get a really nice picture of what was in that environment. Plus, they're alive. So you can freeze that back and go back to that and explore more. So whatever annotation pipeline you use, it was the same story in this approach that we did with those 2 sites. The 2 sites are, you can see the CI, which is the culture independent, and then you can see quasi-metagenomics, QMGS. And in every case, the only time we could talk about the AMR was really with the enriched data. So you could also sort of look at the differential abundance of the clinically important ARGs. And the only plasmids we could annotate were from the enriched data. And when we have annotated the plasmids, we can really sort of talk about the difference between the environment. We're seeing so many trends where there's so much more diversity in the natural environments, as was just mentioned. So the orange ones are connected to the hospital stream, and the blue ones are connected to the drinking water reservoir. So that story is in this publication, if anybody wants to look at that one. And then just to sort of address the One Health, One Data concept, these data sets that we're putting together now, they're archival. They may be libraries, for future generations that want to go back and see what the prevalence of something was. You can look at protists, you can look at everything in these data sets. So intersecting that same data with, we're doing also AMR Bates panels designed by the CARD group, and we also have a Bates panel that we designed for veterinary pathogens and escape pathogens. Thank you. And so that was the concept for the One Health, One Data, but we don't have the panel data back yet, and so I, just conceptually sharing that. Also, as everybody's mentioned, metadata is just invaluable because, if we're going to tie the genomes, the phenotypes to environments, we really need a package that works for that. So this is a package that our group put together. I think that we worked in collaboration. My colleague that does this is Ruth Timmey. She leads this. I believe that I've heard Emma Griffith's name before, but I was very interested in her presentation, which has focused on adding all the attributes that are going to be really important to AMR surveillance and monitoring. So adding that to making sure that ours covers that as well and that those are all interoperable is also going to be really kind of a game changer for this data being valuable. So, and serving the purposes that we hope it will serve and providing the groundwork for the stewardship that needs to occur. So with that, I just want to thank the most important person that I work with is Brandon. He and I are a really small team, but we get a lot done. He's amazing in the lab and amazing with R and all sorts of other things. So, and Heather Tate is sort of emerging as our NARMS scientific director. She's also amazing and wonderful. And Seth Comichaud has been a really valuable bioinformatic resource for us. I will just put this one slide in just to sort of tease you. This is the green boxes are sites that have been sampled. The blue are plasmid types that we're seeing. This was just kind of an interesting, we wanted like an international site, so we did a sampling in Peru, way upriver, where no ballast water gets, kind of in the Amazon and on these unique rivers. And the diversity that we're finding in this environment for plasmids is insane. So we're sort of growing a lot of isolates out of that just to learn. Anyway, and then the ones that are yellow are shared. So I'll leave you with that exciting little piece. I hope to have this data so much better organized by the time we next, have an EDAR and give you an update on what all of this is leading to. So thank you.
But next, I'd like to introduce Kai Shen, who already was introduced by our speaker, David Graham, and she will be talking about evidence-integrated insights on antimicrobial resistance for better understanding One Health.
Okay. Hello, everyone. My name is Tai Chen. Today, I'm going to share our recent work, the evidence-integrated insights on AMR for better understanding One Health. This work aims at using artificial intelligence to interpret the global AMR research. And I will introduce very briefly about the background and more details about the methods and results of the characterizing global AMR research and some future possibilities. So I believe everyone here knows that the increasing AMR is a global health crisis. But AMR may spread among the humans, animals, plants, and the environment. So a One Health approach is very important to handle this issue. One Health is an integrated way to balance and optimize the planetary health. Just like the new One Health joint plan of action said that the data, evidence, and the knowledge is very important for the evidence and for the technical tools and the surveillance systems, which is also same in the global action plan, the objective too, that we need to strengthen the knowledge and evidence base through the surveillance and the research. So we are thinking, how can we make it? What can we do to provide an evidence base and identify the knowledge gaps, especially across the One Health sectors and in a One Health perspective? So the first thing we consider is to find a reliable source. We think maybe the scientific publications is a good choice because, we need to summarize what we have done and then decide what we will do in the future. But the question is, in the age of big literature, there are so many new publications coming out every day. How can we process those massive publications? We think that machine learning gives us opportunities. We can use the methods such as natural language processing to handle the massive literatures. The natural language processing is a computer technology that it will use the computer to comprehend human words, human sentence, and even the scientific publications. So here we are. This is our proposed pipeline for the evidence gathering. It contains 3 main parts. The first part is the database acquisition, which means that we are going to build our own database, which only focuses on the AMR research. It is very different from the, the Web of Science collection and the Superbugs collection. It is only focused on the AMR. And the second part, we will do some quality evaluation of our AMR database and do the filtration to filter the irrelevant publications. And also in this part, we used a machine learning-based way to identify which One Health sector or sectors that one publication is belonging to. So through this way, you know that every publication in our database has its own label. So here comes the third part, which is the most exciting part. So through the natural language processing, we can identify those comprehensive information. Specifically, so the first thing we do is a literature search, and we identify more than 250,000 papers related to AMR, and we use the web crawler to crawl and download the information related to the publications. And the second part, we did the evaluation and the filtration, and to make sure that every publication in our database is relevant to the AMR and is precise. So here is the third part. I will show you more details in the next slide, but here I will highly recommend our advanced searching and retrieval engine. You know that our searching system will take only less than 1 microsecond to extract all related literature and information for a specific term, which means that if you want to conduct your own search about the things that you were interested in, just provide us with your list, your term list, just send email to us, and we can use our search engine through our own AMR database, and we will provide a comprehensive information about all the interesting objectives that you want. And I will give you more details about what we can do. So here is an example. So this is an abstract from a scientific publication. I have done some max on here. You can see that the blue one means the method, and the deep purple means the algae. And you can see that just like the soil can be detected by the natural language processing that it is an environment. And the swim farm also is a kind of environment, the sampling environment. And also the algae and the surveillance methods can also be derived from the abstracts through the natural language processing. So through this way, we used this method to process every article in our database. So we established our own AMR knowledge database. And global foresight and current knowledge gaps of AMR can be characterized by this way, just like the picture in the right side. You can see that we can identify the publications and knowledges across the different income categories and the regions. So I will introduce more results about our AMR knowledge database. The first one is the occurrence of the drug-pathogen combinations in different regions. So you can see that the MRSA is the focus across the globe. And here's another example. The fluoroquinolone-resistant salmonella species were very common in the Asia region, both the West Asia and the East Asia. So you can see that In Africa, the research is very limited, but the fact is the Salmonella species-related infectious disease were still a burden. So this is an example about the about how to identify the knowledge gaps. And this slide is about the ARGs. So you can see that the left picture showed the bubble plot of the ARGs in different countries. The color represents the proportion of one specific ARG detected in one specific country, and the size represents the occurrence of the publications. Just take this one for example, the MCR1 gene, which is in a dark purple in the China, which is consistent with the fact that the MCR1 gene was firstly reported in a Chinese farm. And you can see that the right picture shows the co-occurrence of different ARGs with the pathogens. And after we identify the ARGs across the countries, we are thinking about what kind of surveillance methods did the countries use to detect those ARGs. Just take Iran's example. We found that there are a lot of McA gene-related research in Iran. But what kind of method did they use? We found that more than 80% of the publications in Iran were conducted using only PCR-based method, which is a very traditional way. So we think there is a need for us to identify the methodologies across the globe. So we use our advanced engine to search the methods across the globe, and we find that, oh, that's true, new technologies for AMR research. For example, the metagenomics, the whole genome sequencing, something like that for AMR research seldom occur or are used in the low-income countries, which means that we need to reducing the imbalance in capacity between the low-income and high-income countries. It's very critical to addressing the AMR at a global scale. And then we think about this question in the One Health perspective. And we found that in the environment science, there are a lot of research conducted about the metagenomic methods. But we also find that in human health sector, they always utilize the culture-based methods, which is in large difference between the environmental sector. So we think maybe performing culture-based method and genomic-based method in tandem in all sectors is very crucial for the One Health solutions. And also combined with this slide and the last slide, we think that greater harmonization in research method, both in sectors and both across regions, is very urgently needed. And then we focus more about the One Health research. We firstly use a hierarchical clustering to identify the different development stages of AMR research. But the question is, what are the differences in AMR research within those three stages? So we constructed those complex networks. So, okay, thank you. So you can see that there are intra-sector edges and inter-sector edges. The intra-sector means one publication only focused on one sector. And the inter-sector means that the publication focused on two sectors. For example, if a research was conducted in a hospital and took some samples of the pathogens isolated from the patients, but they also took some samples from the wastewater in the hospital, so these publications will be identified as the human health sector and the environment sector. And an edge will be established through these two sectors to connect each other. So through this way, you can see that with the development of the different stages, the inter-sector research was growing. Maybe this is a good thing. But you can see in more detail that the right picture, it shows the heat map matrix of the between sectors research. You can see that by 2019, the research in the interface between human health and environment were taking hold. But there are still clear gaps in cross-sectoral research, especially linking the plant health and the others. You can see that the plant health with the others interaction is very lighter than other cross-sectoral research. So here is the last slide. You can see that we established our membership-based network of publications in stage 3. And it shows the cross-sectoral research among different regions and among different sectors, and maybe more than 2 sectors. So you can see that there are few publications containing all 5 sectors. Especially in the low-income countries, there were no publications in low-income countries, including all 5 sectors. This is a so informative picture, but I think I'm running out of time. So if you are interested in this picture, maybe you can find me in the post session B. My number is 149. So I will talk a little about our future work. My partner and I, we want to do some artificial intelligence related work to better understand the AMR on the One Health. And we will do the AMR topic chain analysis and mapping the global AMR topics in the future. And we have developed our own chatbot with AMR policy knowledge, which is still unpublished. But if you are interested, you can communicate with us. And we will do more about the AMR knowledge and information sharing. I think it is very important to provide a platform for the general public to better understand the AMR research, especially under the One Health approach. So welcome to contact us. And we really want to take full use of our database and the search engine. So if you want to have future collaborations or use our database, just feel free to contact us. Thank you.